Turmeric is a plant with significant economic and medicinal value as well as its common use as a kitchen spice. This is supported by the fact that Indonesia harvested over 205 million kilograms of turmeric in 2023 based on the data from BPS, highlighting its potential as a valuable resource in scientific advancement, particularly in the health sector. One of the key bioactive compounds in turmeric is curcumin, known for its anti-inflammatory, antioxidant, and anti-cancer properties.
Curcumin offers safer alternative with fewer side effects than in long-term use of NSAIDs that can lead to several health problems, whereas curcumins demonstrate low toxicity and is more compatible with the human body. However, curcumin's potential is hindered by its low bioavailability due to its hydrophobic nature. This results in inefficient absorption by the body, its instability in alkaline environments such as the small intestine further reduces its absorption.
To overcome that hindrance, we proudly present to you Curcumax, Turmeric Capsule Formulation with Microencapsulation technology to enhance curcumin's bioavailability. I'm Diva.
And I'm Kaila, and we're from
Satuan Bangsa School, Indonesia.
This research examines the effect of microencapsulation in curcumin's bioavailability on turmeric capsules. We compare its effectiveness with conventional formulations in order to optimize curcumin release for better efficacy. To make the microcapsules, we begin our experiment by maceration, followed by a rotary vacuum evaporator to concentrate the extract, and a characterization test of curcumin quantitative content using UV-based spectrophotometer and moisture content test.
Next, microcapsules are formulated using different compositions. These microcapsules are then tested for their physical parameter as well as particle size using scanning electron microscope. Finally, an in vitro test is conducted under gastric and intestinal conditions to evaluate their performance.
The main components of our encapsulant are maltodextrin and cassava starch. We use 3 different formulas. The first formula, we use 3% of cassava starch and 1% of maltodextrin. The second formula, we use both 2% for the cassava starch and 2% for the maltodextrin. And for the last formula, we use 1% of cassava starch and 3% of maltodextrin. The turmeric extract has good quality according to the Indonesian Herbal Pharmacopoeia. The moisture content of our turmeric extract was determined to be 2.3%, which is important in order to lower the risk of microbial and fungal growth. And for the curcumin content, we got a number 12.4%, which passed this standard.
So the result of the microcapsules, F3 produced the most volume, and we can say that the more maltodextrin contained, the more volume produced, as F3 have the most maltodextrin. The yellow pigment in the microcapsules represent the curcumin. So from F1 to F3, the curcumin content is decreasing. However, as we can see in the table there, the size of the microcapsule is decreasing from F1 to F3. This results that the granules are relatively round, indicate that the active ingredient was successfully encapsulated. Smaller granule size can enhance the bioavailability of the active compound by increasing its dissolution rate in the body.
And here are the results for the in vitro test. During gastric condition, the bioavailability increased from F1 to F3, meaning F3 with 3% of maltodextrin is suitable in acidic condition with pH level of 1.4. While in intestinal condition, the bioavailability is the complete opposite from the previous one, which decreases from F1 to F3, meaning F1 with cassava starch dominance is suitable in higher pH condition.
Based on our experiment, we can conclude that Formula 3 is the most optimal formulation due to its superior bioavailability in acidic gastric condition, reaching to 1.4%. This suggests that F3 exhibits a greater stability in acidic gastric condition and retains a higher amount of curcumin compared to other formulations.
Although F1 demonstrates the highest bioavailability in intestinal condition, the morphological structure of F3 is more favorable due to its particle size 10 micrometers smaller. Smaller particle size contributes to better solubility, enhancing its overall effectiveness.
So, considering both bioavailability and particle morphological structure, F3 is identified as the most suitable formulation. To improve this research, we recommend conducting a stability testing to assess the shelf life of the microcapsules and studying how the digestive system affects the curcumin release. These steps will help to develop a more stable and more effective curcumin formulation.
And that's a wrap on our project. Thanks for watching, and don't forget,
Curcumax! Tiny particles, big impact! Bye everyone!